Good morning students. In this video we are going to discuss about endomembrane system from the chapter Cell, the Unit of Life, and let us see today's concept in detail. While each of the membranous organelles is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their functions are coordinated with each other. Understand? So especially the endomembrane system includes the first cell organelle, endoplasmic reticulum, shortly called as ER, Golgi complex, it is also called as Golgi bodies, and third one lysosomes, and fourth one vacuoles. All these four cell organelles are membrane bound structures which are present in the cytoplasm. So as we learned, cytoplasm is the center for all cellular activities. It is also called as arena of the cell. In this cytoplasm, which is in a fluid matrix structure, these four cell organelles are presented in different locations and performing different functions but coordinating with each other. Structure is different, shape is different. Each cell organelle has its own functions, but each cell organelle is coordinating in function wise. That's why scientists made one unit about these four cell organelles. Right now, there are many cell organelles are there in the cytoplasm in both plant cells, animals like mitochondria, chloroplast, peroxisomes. They are not coordinating with each other and their component is also different. So these cell organelles are not at all coordinating with each other, even though they are different in structure and shape, even though they are considered as a major part of the cells, but they are not included under the endomembrane system. They are not considered as a part of the endomembrane system because those functions are not coordinating with the cell organelles which are presented or included in the endomembrane system. So the main criteria, what is the criteria here, is nothing but the cell organelles functions are coordinating with each other, even though their structure and shape is different, even though each cell organelles are different, but they are coordinating with each other. Those four cell organelles can be seen under endomembrane system on the screen right now. Now I am going to explain in detail about each cell organelle under the unit of endomembrane system. Okay, now let us discuss about the one important point here. All the four cell organelles on the screen you can see very clearly numbering 1 2 3 4. All these are included under the endomembrane system. What are those cell organelles? Endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. Understand? So now I am going to explain about the first cell organelle under the endomembrane system, that is endoplasmic reticulum. You can see the diagram on the screen. Let's talk about it. Electron microscopic studies of eukaryotic cells reveal the presence of a network or reticulum of tiny tubular structures scattered in the cytoplasm. They are called as endoplasmic reticulum, shortly called as ER. There are very tiny tubular structures present in the cytoplasm. As we discussed in the earlier videos, electron microscopic adventure and usage have delivered in the decade of 1950s. So this electron microscopic studies of eukaryotic cells reveal the presence of network or reticulum structures in the cytoplasm which are present in different sites of the cell. Those are called as endoplasmic reticulum. Again, let us see some information in detail regarding this endoplasmic reticulum. This endoplasmic reticulum divides into two main parts, intracellular space, into two distinct compartments. What are those? The first one is luminal inside the ER, and the second one extraluminal towards the cytoplasm. So these are the two compartments can be seen in the endoplasmic reticulum. You can see on the diagram very clearly mentioned here that violet color spherical structure is nucleus covered with a membrane called as nuclear envelope. Outer nuclear membrane, inner nuclear membrane, together called as nuclear envelope. So from the outer nuclear envelope, from the outer nuclear membrane, the endoplasmic reticulum is emerging or forming. Understand? So the endoplasmic reticulum divided into two categories based on the presence of ribosomes. Now let us see what are those. So the endoplasmic reticulum, which has ribosomes on the surface, it appears rough in nature. So the endoplasmic reticulum is called as rough endoplasmic reticulum. Pink colored structure on the diagram. Next, the endoplasmic reticulum, which do not possess ribosomes on the surface, they appears very smoothly, since they are called as smooth endoplasmic reticulum. Both the parts you can see in the diagram. As I told you, ER divides into especially intracellular space divides into two categories. One is inside the space, second one is outside the space. Outside the space means towards the cytoplasm. Assumes the total white colored structure is a cytoplasm. Into that cytoplasm, the space which is projecting or emerging, that is called as extraluminal. As we know that lumen is a free space, right? So inside the ER, some space called as, it is called as intraluminal space. Then outside the lumen, it is the space is called as extraluminal space. Understand? Now one more important point I would like to repeat here. The ER often shows ribosomes attached to their outer surface, right? The endoplasmic reticulum bearing ribosomes on their surface is called as endoplasmic reticulum, shortly called as RER, right? Then in the absence of ribosomes, they appears smooth and are called smooth endoplasmic reticulum, called as shortly SCR. Now one important point I would like to tell you here. Smooth endoplasmic reticulum and rough endoplasmic reticulum, their functions, I just wanted to bring to your notice. You can see in the diagram on the screen, the smooth endoplasmic reticulum do not possess any kind of structures on the surface, mainly ribosomes. So ribosomes are not attached or not present on the surface of the endoplasmic reticulum, since it is smooth in nature, called as smooth endoplasmic reticulum, right? So the smooth endoplasmic reticulum is the major site for synthesis of lipid. In animal cells, lipid-like steroidal hormones are synthesized in SCR only. This is very, very important for NEET examination and other competitive examinations also. Please make a note in the notebooks. I repeat once again, on the screen please watch the diagram. Smooth endoplasmic reticulum and its functions. The smooth endoplasmic reticulum is the major site for synthesis of lipid, and in animal cells, lipid-like steroidal hormones are also synthesized by SCR only, or in SCR only, smooth endoplasmic reticulum only. Hope you understood this point, right? Next, I am going to explain about rough endoplasmic reticulum, shortly called as RER. So this RER directly originating from the outer nuclear membrane, very closely pressed to the nucleus, Make sure this point must be clear to everybody. You can see very clearly in the diagram. So the violet colored structure is nucleus. From the nucleus, rough endoplasmic reticulum is emerging, especially from which membrane? Outer nuclear membrane. So on the outer nuclear membrane, you can see small spherical structures. Those are called as ribosomes. Since the surface is appearing like a rough nature, the endoplasmic reticulum is called as rough endoplasmic reticulum. Understand? Now let us see. This RER is frequently absorbed in the cells actively involved in protein synthesis and secretion. Understand? So please make a note of this point. This is also very important for competitive examinations. I repeat once again, rough endoplasmic reticulum is frequently absorbed in the cells actively involved in protein synthesis and secretion also. So they are extensively and continuous with the outer membrane of the nucleus. Understand? So this point is also very important. So there are extensions and continuous parts of the outer nuclear membrane which is covered to the nucleus. Understand? So students, so hope you understood this one. You can see very clearly in this diagram on the screen, which you can able to see right now. So the spherical structure is nothing but called as nucleus. It has two layers, inner nuclear membrane, outer nuclear membrane. It also has ports called as nuclear ports, and from the outer nuclear membrane which is emerging endoplasmic reticulum. Understand? So the structure can be seen very clearly on the screen in this diagram. Again, rough endoplasmic reticulum and smooth endoplasmic reticulum. These two are the parts in the endoplasmic reticulum. Understand? Now students, let us learn about another important cell organelle in the endomembrane system, that is nothing but Golgi body. It is also called as Golgi complex. On the screen you can see the diagram. In the diagram they showed as green colored structure. Understand? Now let us see about one great scientist here, Carmelo Golgi. In the year of 1898, first observed densely stained reticular structures near the nucleus. This point is very important for NEET. Please make a note. I repeat once again, scientist name Carmelo Golgi. In the year of 1898, first observed densely stained reticular structure near the nucleus. These were later named as Golgi bodies after his death, after him. Understand? Since he discovered with the scientist name only, those structures or cell organelles are called as Golgi bodies as his memory, as his remembrance. Next, they consist of many flat disc-shaped sacs or cisternae of sizes 0.5 μm to 1.0 μm diameter. You can see on the screen. Cisternae has one specific characteristics. These are stacked parallel to each other. You can see on the screen. Stacked parallel to each. So each one vary from one to another. Varied number of cisternae are present in a Golgi complex. Right? So the Golgi cisternae are concentrically arranged near the nucleus. Where they are location near the nucleus, concentrically arranged near the nucleus with the distinct convex face called as cis or the forming face. Then concave shaped trans or maturing face. Understand? So now this point is also important. I repeat once again. The Golgi cisternae are concentrically arranged near the nucleus with the distinct concave and convex shapes structures. Now let us see one by one. The convex one is called as cis or forming face, which is very close to endoplasmic reticulum. Next, the concave shaped structure is called as trans phase. It is also called as maturing phase. Understand? Now, one more important point regarding this one. The cis and trans phases of the organelle are entirely different but interconnected with each other. You can see how many cisternae are there. Each structure is called cisternae, but here you can see on the diagram 1, 2, 3, 4. So 4 cisternae are present. They run parallel to each other, has two phases, cis and trans. Cis phase is forming phase, trans phase is maturing phase. Cis phase is close to the endoplasmic reticulum, that too rough endoplasmic reticulum. But even though organelles are entirely different but interconnected with each other. Understand? Now, one more important point regarding this Golgi complex. So, the Golgi complex or Golgi operators are principally performs the function of packaging materials. This is very important. What is the function of the Golgi complex? Packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Understand? So materials to be packed in the form of vesicles from the ER fuse with the cis phase, since the cis phase is attached to the ER of the Golgi operators, and move towards the maturing phase. The maturing phase is called as trans phase. So from cis to trans phase, cis phase to trans phase. This explains why the Golgi operators remains in close association with the endoplasmic reticulum. Why they're closely associated with the endoplasmic reticulum? Because they are connected with the function wise. Understand? So especially the rough endoplasmic reticulum has ribosomes. Those ribosomes are meant for synthesis of proteins. Those proteins are receiving by these Golgi operators, and they're packing these protein materials, and those packed protein materials are entering through cis phase and maturing in the trans phase. Then what may happen here? Again, a number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi operators before they are released from its trans phase. So releasing from which phase? Trans phase, and entering from RER, which through which part? Cis phase. But releasing into the cytoplasm or out of the cell, it is nothing but through trans phase or maturing phase. Golgi apparatus is the important site for formation of even glycoproteins and glycolipids. This is very important one. I would like to show one more important diagram. Please watch this diagram. So this Golgi complex is also important site for synthesis of glycoproteins and glycolipids. This is competitive part, competitive question, most important. Please make a note. So in this diagram you can see cis phase, which is present at the top, trans phase, which is present at the bottom. So light yellow color structures are nothing but incoming transport vesicles. They are nothing but protein substances through cis phase. Cis phase is also called as forming phase, connected to rough endoplasmic reticulum RER and coming into the lumen free space of cisternae. Understand? Again, in the lumen, what may happen? Newly formed vesicle are releasing or secreting into the cytoplasm. You can see in this diagram, right side one vesicle is emerging out, coming out of the lumen. From which phase? Trans phase, which is also called as maturing phase. Understand? Next, students, I would hope you understood about Golgi operators. Now I am going to explain about third important cell organelle. It is nothing but lysosome. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. See how the creation. So these lysosomes are associated with the functions of Golgi operators. How they are forming? I repeat once again. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi operators. Right? So the isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes. This is very important one. Please make a note. Important for competitive examinations. The isolated lysosomes vesicles have been found to be very rich in almost all types of hydrolytic enzymes. Then let us see what are those hydrolytic enzymes. You can see the structure of lysosome on the screen. So this lysosome is rich in hydrolytic enzymes. What are those? Hydrolysis. What are those hydrolysis? Lipases, proteases, carbohydrases. Understand? So now let us see the functions in detail. Lipases helps in digestion of lipids. Proteases helps in digestion of proteins. Carbohydrates helps in digestion of carbohydrates. So optimally active at the acidic pH. This is very important. Please make a note. So all these hydrolyses are very active at acidic pH. These enzymes are capable of digesting carbohydrates, proteins and lipids, and even nucleic acids also. What are the nucleic acids? DNA and RNA. Please make sure that this point must be understand to everybody. Understand? Now students, I would like to tell you one more important part. It is nothing but vacuoles, last cell organelle in the endomembrane system. You can see the structure of a centrally large space vacuole in the plant cells. So these vacuoles are present exclusively in plant cells, whereas they are absent in animal cells. Understand? So this is exceptional case. So now let us see about vacuoles in detail. The vacuole is the membrane bound and space found cell organelle in the cytoplasm. So I repeat, membrane is present on the surface of the cell organelle, and as well as space can be seen in the cell organelle. Understand? This cell organelle is present in the cytoplasm, and it contains water, sap and excretory products and even other materials also, which are not useful for the cell. Understand? So since the free space is present in the vacuoles, they are helpful for storage of water, sap, excretory products and other materials also, which are not so useful for the cell. Understand? So the vacuole is bound by a single membrane called as tonoplast. This is very important. I will show you tonoplast structure in another diagram. Please go through it. Yeah, in this diagram you can able to see very clearly tonoplast structure. So a single membrane is covered with vacuole. The single membrane is called as tonoplast. This is also very important for competitive examination. Please make a note. Next, one more important point regarding these vacuoles, and especially in plant cells, the vacuoles can occupy up to 70% of the volume of the cell. How great it is, right? So 70% of the volume in the plant cells are occupying by the vacuoles only. Understand? Next, one more important point. Let's discuss about vacuoles. In plants, especially, the tonoplast facilitates the transportation of the number of ions and other materials against concentration gradient into the vacuole. So this is the important function. So in plant cells, tonoplast facilitate the transportation, right? So number of ions and other materials against concentration gradient into the vacuole. Hence their concentration is slightly or significantly higher in the vacuole than in the cytoplasm. So in the cytoplasm, the concentration or the concentration is very how it is different. How it is different? Significantly higher in the vacuole. So the concentration is much higher in the vacuole than the cytoplasm. So in the cytoplasm only, less concentration can be seen in the ions and materials, whereas comparatively with the vacuoles. Understand? So in the vacuole, the concentration gradient is much higher. So helps in transportation. Again is to concentration gradient. So again is to concentration gradient means from lower concentration to higher concentration, or higher concentration to lower concentration, right? Now, very important point, hence their concentration is significantly higher in the vacuole than in the cytoplasm, especially in some organisms like amoeba. In amoeba, the contractile vacuole is important for excretion. This is very important point, please make a note. In amoeba, a microscopic organism, right? So in this amoeba we can able to see one specific nature. It has one vacuole. The vacuole is called as contractile vacuole. It helps in excretion. It stores the excretory products. Then in many cells, as in protists like euglena, food vacuoles are formed by engulfing the food particles. This is very important. Please make a note. So protists, example euglena. So in many cells like in protists, example euglena, food vacuoles are present because they can able to synthesize food by themselves. Understand? So this is because euglena is also called as mixed trope. When there is a huge amount of or well amount of light, it can able to perform a photosynthesis process and synthesizing its own food material like a autotropic organism. Then when there is a deprived or poor amount of sunlight is present, then it can depend on other lower organisms as a predator for food as a heterotropic organism. Since it is exhibiting autotropic nature and heterotropic nature, it is called as a mixed trope of the kingdom Protista. So in these protists like euglena, food can be stored as a vacuole called as food vacuoles. These food vacuoles are forming by engulfing the food particles. Understand? So this is about vacuoles. Students, hope you understood about endomembrane system. With this I conclude this session. In the next session I will explain about other cell organelles, mitochondria, plastids and etc. Thank you for your watching.